In the second part of the video, we're going to talk about planning of movement. Where does it actually happen in the brain? How do we plan how to move? So the cerebral cortex is going to decide about what kind of movement will happen. Where is the body right now? Where does it need to go? And exactly which muscles need to be contracted, which ones need to be relaxed, and how do we need to move the body to get into where we're going. So where are the planning areas? We already know about the M1, the motor cortex 1, which is right next to the S1, which is the sensory 1 cortex. Okay, so those two strips with their representation of the entire body, and we'll look at it again to remind you what it looks like. The M1 is also known as area 4 of the brain, the Broca area 4. In addition, to execute a movement, we need more areas, we need more coordinated cortical activity. So two areas in the frontal lobe are supplementary motor area, SMA, within area 6, and the premotor area, PMA. And I'm going to point those out to you. The posterior parietal complex, these are areas which will receive some of the input from the occipital lobe, from the visual cortex. So they're going to be involved in coordinating what we see with where we're going, how we're going to move. So it's not surprising that these areas are going to heavily participate in the decision making that goes on in the prefrontal cortex as to how to actually direct the willful movement. So the primary motor cortex, this is going to be more or less direct control of muscles, and we'll talk about exactly how that happens a little bit more, but not too much more, because these are going to be the pathways that we have just talked about, the spinal pathways coming down from M1 and going down the cortical spinal pathways. Area 6 is a higher motor area. Things are going to happen in area 6 before they happen in M1. This area 6 is going to be where the decisions have to be made, and exactly what neurons will need to be activated, how the movement will be executed, then that information will be relayed to M1, and then from M1, from the motor cortex down spinal pathways into the actual muscles to execute the function. Let's look at the brain and the areas involved. So here we have M1, here we have the central sulcus, and directly opposing that is the S1, sensory 1, so that you can integrate information from the sensory area and relay directly to M1. And here is the premotor area and the SMA area, part of the frontal lobe and the prefrontal cortex. Here is the posterior parietal complex, really adjacent very closely to the occipital part of the brain, which is receiving the visual information. So those areas are all going to be involved in planning movement. Now let's talk about the scientists that really made major contributions to our understanding how the motor cortex is organized and how the decisions are being made. This is Penfield, and he was a brain surgeon, and he actually is credited with creating the homunculus maps of the motor cortex. So what he was doing is he was actually operating on epileptic patients, so people who had epilepsy, seizures, and he wanted to see whether there was a way to find an area that was actually causing seizures and whether he could just simply cut it out. This is what they wanted to do. They wanted to cut out the area of the brain that was causing seizures. In order to make sure that he didn't cut out anything very, very important, he would keep his patients awake during operations so that they could tell him what they were experiencing. And remember, there are no pain receptors in the brain. So these people had electrodes stuck to their cortex, but they didn't really feel any pain. But what they did feel, Penfield was actually able to trigger activations of muscles sequentially in all different parts of the body, and he was very clever. So he would actually, every time he did it, he would make a map of what was happening in the brain, and he was watching what was happening in their bodies, how their bodies were responding from the activation from the motor cortex, electrical activation. And so he was actually able to come up with a very good mapping. And this is a picture from 1958. This is how the surgeries were happening. Here is the patient. We can see his face being held by the hand by a nurse and another practitioner, and Penfield is operating on the brain of a patient. And there is EEG monitoring the function of the brain. So this is the result of these somewhat dramatic procedures. This is the mapping of our motor cortex, and you can see again areas of the motor cortex representing different parts of our muscular system. So heavily represented is the hand, the face, the tongue, less heavily represented the wrist, elbow, shoulder, trunk, and so on. So there is this distortion, disproportionate amount of space in the motor cortex devoted to these particular parts of the body. Here is another way of mapping the brain. This involves putting a set of electrodes on top of the brain of a conscious patient. So this is a living brain right here that you see, flaps of skin pulled away and the skull open. And here are the electrodes, and can very carefully stimulate the brain activity and watch what happens, and perhaps cure the patient, perhaps find where the lesion is in the brain. So now, what was learned from this? That the area 5, so let's find area 5. Area 5 gets inputs from area 1, 2, and 3. So those are all the S1 areas and the areas adjacent to S1, so the sensory areas. And then area 7 gets input from higher order visual cortical areas. Remember, we talked about the MT areas where the processing of movement happens in the visual neurons. So this is one of the areas that is contributing here to the motor functions. Now, the frontal lobes, those are the areas that are involved in decision making, really, and where we actually analyze what's going to happen if we do A versus B. So consequences of an action. And then area 6, once the decision is made, this is where it is converted into specific action and specific sequence of movements, and then that command is sent to M1, and M1 is going to direct the action of neuron. Interesting experiments were done by Per Roland, and what he did was he did PET scanning. So remember, this is positron, using positrons, and essentially labeling the brain with positrons and looking at the activation of different areas of the brain. The interesting experiment was that he asked the subjects to learn a series of finger movements, and then he would be scanning their brain and asking them to repeat that movement from memory and looking at which areas of the brain were activated. So as you would expect, you have activation of somatosensory because you have to have the feeling of your hands, the proprioceptors coming in, moving the fingers. Parietal posterior, not surprisingly, also trying to imagine that movement first before it happens. Part of the prefrontal cortex, decision making to move the fingers, and then area 6, finally area 4, and then involvement, direct involvement of the neurons. But what was even more interesting is he then did an experiment where he only asked the subjects to think about the movement, the same series of movements, without actually moving the fingers. And all these areas lit up except for area 4. And again, to remind you, area 4 is the M1 cortex. So there was no need to actually execute the movement, but the brain had thought of making the movement. Area 6 was involved, but area 4 was not. Second really important set of experiments was done on the monkeys. And these were experiments on, actually in the monkeys, they were able to read directly from the neurons. So these readings are from the PMA area. Let's look at where that is. PMA is part of the area 6. This is the premotor area, directly adjacent to area 4. So now they're reading from individual neurons in PMA. So this isn't a complete drawing of what's happening. This monkey also has an electrode stuck in its brain. And the monkey has been taught to anticipate a stimulus. There's 4 possible stimuli that can light up. So this is sort of a ready, set, go scenario. If we read from the PMA neuron, the monkey is just waiting for something to happen. The neuron is just ticking along. The instruction stimulus comes on, so this particular window lights up red, and the monkey knows that now it has to get ready. It has to get ready to trigger for the trigger stimulus, which is lighting up in blue in a moment. The monkey knows that when the red window lights up, the next thing that will happen, the blue button will light up and the monkey will press the blue button and then get a reward. So if we look at the activity of the neuron, the PMA neuron, premotor neuron, gets activated as soon as the stimulus comes in. But once the stimulus is on and the monkey actually executes the movement, that neuron stops firing. It is no longer needed because now the information has been passed on to M1. In addition, there's some really interesting neurons again, they're all in area 6. The neurons will fire when the subject is only looking at the movement. So this was again done in the monkeys, reading directly from specific cells. These neurons are called the mirror neurons. So the monkey is looking at something happening and the brain is mirroring exactly what's happening. So here we have the actual experiment. Reading from the mirror neuron, monkey is picking up a peanut and we can see a certain pattern of activity. Now, the monkey is not picking up the peanut. The monkey is watching another monkey pick up the peanut, while its brain is behaving as if it is the monkey herself picking up the peanut. The monkey, again, is watching a human pick up a peanut. Those mirror neurons can mirror that action again. But here we have something interesting. A control. The human uses forceps to pick up the peanut. The monkey cannot relate to picking up something with forceps, so it does not have mirror neurons that can execute that movement. So it's just watching and that neuron stops firing. Okay, so mirror neurons will only fire in response to something, some imaginary movement that can actually be recreated in the brain of the subject. Okay, we will take a break, short break here.